It's a pleasure to have you here. We're starting our outreach to the community again to tell you about all of the exciting things that are going on at Scripps, and this is the first in a series of activities that we'll do to do that. I was asked to lead off this lecture series, but I'll be followed by a number of younger and more dynamic colleagues who are on the slide here. What I was asked to talk about is really the vision for Scripps and where we're heading here in the future. And really, when I took on this position a couple of years ago, thinking about biomedical research institutes, the question arises whether we really need a new model or to revise the existing model for what we do in nonprofit biomedical research. And traditionally, we've done scientific discovery, and Scripps has a rich history of scientific discovery. But maybe it's time not only to make those discoveries, but to internally accelerate the translation of those scientific discoveries to new medicines. And by doing so, you can have a bigger impact on the public welfare, which I think is what many of the public who fund science expect, but you also may be able to create a new mechanism for funding basic research that augments the federal funding of research, which those dollars continue to dwindle. So what we really want to do is create a model where we not only continue to make great discoveries, we accelerate the translation of those discoveries to new medicines, and by doing so, we enable a self-sustaining model for biomedical research where we create new resources to continue to invest in research and drug discovery. Just a little bit about Scripps, for those of you who aren't familiar with Scripps, also known as TSRI. We may be one of the best-kept secrets in San Diego. If you're here to hear about whales, you have the wrong Scripps, okay? As Jeff said, we have about 180 faculty, about 2,000 staff, grad students, and postdocs. We're one of the largest biomedical research institutes in the United States. Distinguished faculty and board. We actually have a graduate school that started 26 years ago, and despite its small size, it's ranked in the top 10 in chemistry and biology, and actually number 2 in biochemistry. And I think to some of our surprise, the premier scientific journal Nature did a survey of all scientific institutions around the world that have had the biggest impact on scientific innovation, and Scripps actually came out number 1. So it's got a long history of scientific excellence. And that history bridges physical sciences like chemistry to neuroscience to immunology to structural biology and molecular medicine. And it also bridges, as Jeff alluded to, the development of new medicines. And I think, Scripps has contributed 50 or 60 different companies to the San Diego economy. Just briefly, some of the highlights of Scripps Research is our neuroscience group is very focused on sensory neurobiology. How do you sense external stimuli, and how are those stimuli translated to effects on brain development, and behavior? And one example of that is asking, how do you as a person sense temperature? And how do you sense mechanical force? And how are those stimuli converted to electrical signals via ion channels that then are signaled to the brain? And so Scripps has really been at the forefront of understanding the structural basis for sensory perception. In immunology, the focus of the Scripps research is both to understand and control the immune response. And so how does your immune system, even though it may never have seen a virus that impacts you, know to respond to that virus as a foreign substance that it has to eliminate from the body versus attacking self? And the outcome of those two things is very different. One is it prevents infection, and the other is it causes autoimmune disease. So Ian Wilson at Scripps really made a huge discovery of the molecular basis for how we distinguish foreign versus self molecules in the T-cell receptor MHC complex. And we also have people who are interested in stem cell biology and reprogramming cells. Kristen Baldwin is taking fibroblasts from patients, skin cells from patients with neurodegenerative disease, and turning those skin cells into diseased neurons in a dish that now you can study Alzheimer's or Parkinson's in the lab in a simpler system, and you can actually treat those cells with drugs to see if you can reverse the disease process. And we have a large chemistry group that really is focused on understanding the chemical reactivity of big and small molecules, and how do you make molecules with incredible efficiency and selectivity, which is really important today when we worry about, green chemistry. And both developing chemistries for small molecule synthesis, and Barry Sharpless, who's our resident Nobel laureate, developing new chemistries to modify large proteins and DNAs with exquisite selectivity. And we also have efforts in synthetic biology. All forms of life that we know have a 20 amino acid genetic code. And if you discover life on another planet, what if it has a 21 amino acid genetic code? Will that have an evolutionary advantage? So we're asking the question, what would we look like if God had worked on the seventh day and kept going, okay? And so Scripps is leading this effort in creating new organisms with new codes. And as Jeff alluded to, Scripps has created a lot of new medicines. Surfactant for infant respiratory distress syndrome. Charlie Cochran. The Lerner Lab created phage display technology that helped make Humira, which is a widely used drug for autoimmune disease. Jeff Kelly was a pioneer of a breakthrough drug that just got breakthrough status from the FDA for transthyretin cardiomyopathy. And ozanimod should soon be approved for the treatment of MS and IBD. But despite all of these discoveries, and new medicines, there's an overwhelming need for new medicines, Degenerative diseases and diseases of aging, we really don't have cures for these diseases. There are a huge number of childhood diseases. Type 1 diabetes, we don't have a cure for. Autoimmune diseases, neglected diseases like malaria and TB that kill more people in the world than any other disease. We don't have cures for those. And cancer. We really don't understand how to cure cancer. So that's the real challenge of the future. There's a huge unmet need. And why does that still exist given the discoveries that we're making today? And part of the reason for that is because a lot of those discoveries are made in basic research institutions like Scripps and Salk and UCSD and others. And all those discoveries are made there, those institutions in general don't have the infrastructure, expertise, and know-how to convert those discoveries into drugs. They have to be partnered with the pharmaceutical industry. And there are intrinsic barriers between the way the pharmaceutical and biotech industry work and academics work that create, logistical roadblocks that slow the whole process of turning new discoveries into new medicines. And this ineffective coupling of basic research and the commercial life science sector really is a challenge. And so how do we solve that challenge? So a few years ago, while I was running my lab at Scripps, we thought about that, and so we formed another nonprofit institution called Caliber. It's a 501c3. And we formed that based on an experience I had when I moved to Scripps, where we were funded by the Novartis Research Foundation with roughly over 10 years, $1 billion, to develop new tools for drug discovery and use those tools to make new drugs. And so we developed a huge number of technologies that accelerated drug discovery, learned how to use them, and it was a billion-dollar education, which is a rare opportunity. And based on that education, the idea developed whether you could actually do drug discovery effectively in a not-for-profit sector without some of the constraints of, the financial constraints and commercial decision-making processes of pharma. And so we started Caliber, and Caliber grew. It was funded by various pharmaceutical companies, by foundations like the Gates Foundation, Wellcome Trust, JDRF, and others. And it created an infrastructure and expertise for doing drug discovery in a nonprofit setting. And I'll tell you a little about some of the drugs and infrastructure that were developed. But based on what we did at GNF, we recreated all of the kinds of tools and capabilities and infrastructure that one sees in a large biotech or pharma. High-throughput screening, protein engineering, medicinal chemistry, pharmacology, and even a clinical team for IND-enabling studies and clinical research. We also brought in a lot of the tools that we developed at GNF. If I could have the movie, great. I can do it myself. So if you want nowadays to make a cancer drug, what you can do is take a cancer cell that you'd like to kill, put it in a little well, and add a molecule and see if you kill it. And then you take a normal cell and you add the same molecule to a normal cell and you see if you kill it. And if you selectively kill the cancer cell and not the normal cell, you're on your way to potentially doing a cancer drug. The challenge when we started this was technologies that were created in academic institutions in places like Scripps allowed you to make libraries of 1 million plus 2 million molecules. But every well you put that molecule in costs $1 to screen. So if you did an experiment with 2 million molecules, it costs $2 million. And it took about 3 weeks. So if you have 10 experiments that you can do, and they cost that much money and take that much time, you sit around forever thinking about which is the best experiment to do. The better thing to do is I recruited my old college roommate who was a chief engineer at GM, and I said, we got to make this work cheaper and faster. So he brought in all the automated robotics from GM and built all of these high-throughput systems that you now see throughout the world. And we reduced the cost to a couple of pennies a well and reduced the time to 2 days. So all of a sudden, instead of saying which one of these 10 ideas is the best idea, you do them all. All of a sudden, the risk scenario changes because you're prepared to do high-risk experiments because they don't cost you any money and any time. So using technologies like this, we have imported those in, and now we have those at Scripps and Caliber. And the other essential part of the model is to collaborate. The idea that even as an institution like Scripps and Caliber, you can understand all of the biology that impacts all of the diseases you're working on is naive. What scientists really have to do is collaborate with other folks who have complementary skills and know-how and knowledge. And so we've created a very, very collaborative model to do translational research. So having created Caliber and having Scripps already existing, the idea is, can we create a new model for basic biomedical research where we bring together the talent, creativity, and investment in basic biomedical research that exists at Scripps and combine that with the translational and drug discovery capabilities of Caliber. And by bringing the two institutions together, go all the way from a discovery at the bench to the patient bedside. And so that's the idea that we'd like to create as a nonprofit biomedical research. And if we can do that, we will certainly accelerate the development of new medicines for all those unmet needs that I described. And a lot of folks have tried to do this and build drug discovery capabilities within an academic institution, and that's hard because the culture and methods and processes that you use for drug discovery are very different than that in academic research. Academic research is by and large a cottage industry. Every faculty works primarily on his or her own. Drug discovery is teamwork and very Darwinistic. So if something doesn't work, you stop, okay, because it's very expensive. So it's hard to build a drug discovery culture in an academic setting because I don't know if any of you have ever tried to tell a professor what to do, but it just doesn't work, okay? Especially if you're telling them their idea is not so good. So I think the advantage we had is we simply bolted two existing organizations together to create this capability. And as I said, if we can move all the way from a discovery to a molecule in patients, the value creation, not only in terms of the impact on public health, but the commercial value creation is huge. And the research institution will capture a far greater part of that value versus the shareholders and stockholders and so forth. And you can reinvest those revenues back into the research enterprise and really fundamentally change, I think, the resources you have available in the nonprofit sector to do great science. So that's the model, and will it work? So it's a big experiment, and we have a terrific group of people who are enthused about this. And so I'd like to begin to tell you about our progress towards this goal, making our vision a reality. So let me tell you some vignettes. And I'm not going to go into the details, just the 30,000-foot view. And one of the programs we're working on is prostate cancer. About 1 in 10 men. It's the second leading cause of cancer in men. About 30 to 40% of those patients get a relapse. And if you get a relapse and it metastasizes, there are really no good treatments for that. So how do you treat metastatic prostate cancer? So the exciting technology now and approach in cancer biology is to recruit the immune system itself to kill cancer cells. Normally you think about the immune system as killing virally infected cells or microbes. People have figured out how you can use your immune system to destroy cancer cells. And so in the case of prostate cancer, what you'd like to do is recruit a cell in the immune system called a cytotoxic T cell to the cancer cell and use the T cell to kill the cancer cell. So how do you bring those two together? It's really a simple idea. You make a molecule that has two ends. One end grabs onto a protein highly expressed in prostate cancer cells. It's very related to PSA. The other end grabs onto a protein that is expressed on the surface of the cytotoxic T cell. So you have a little bridge, adapter molecule, that brings these two together, brings the T cell right next to the cancer cell, and proximity kills the cancer cell. And the T cell doesn't care about drug resistance. So does this work? Well, we went over to UCSD and got patient-derived cells from metastatic prostate cancer that have metastasized to the bone. And in an orthotopic xenograft model, if you look at the growth of the tumor, it's really quick. If you actually treat with that molecule I showed you on the previous slide with human T cells, you absolutely ablate the cancer. So we're actually looking at putting this into the clinic next year with funding from Wellcome Trust, and we think this could be a first major therapy for relapsed metastatic prostate cancer. Now, the other approach people are taking to using the immune system to kill cancer is to actually take the T cells out of your body, genetically re-engineer those T cells with an antibody that's expressed on the surface of the cell and targets that cell to the cancer cell. So you engineer the T cells, you put them back into the body, and all they want to do is home in on the cancer cell and kill it. And these are called CAR T cells. And what's amazing is people are seeing complete cures. They're also seeing patients die, okay? And so the challenge there is you can't really expand this therapy to a huge number of solid tumors because of the safety issues. And really you can only use it in healthy young adults right now. And why is that? It's because it's a living drug. And when you put it back in the patient, you can't control it. And if I have a headache, I'll take 2 Advil, I'll take 3 Advil, I'll take 4 Advil. I can just dose titrate the Advil, okay? With this, you can't control it. So in some cases, the T cells expand so quickly they kill the patient. So what you really want to be able to do is dose these cells the same way. So we use the same concept. We use an adapter molecule. And this adapter molecule is a protein. On one end, it has a little piece of protein that binds to the CAR T cell, the killer cell. And on the other end, it has a piece of protein that binds to the tumor cell. Now, if you put these CAR T cells into the patient, they don't do anything because they don't see anything. If you put in this adapter molecule, it bridges the CAR T cell again to the malignant tumor cell, but you can dose titrate. So if you start out with a huge tumor burden, you put in a small amount of, you put the CAR T cells in, they don't do anything. You put in a small amount of the adapter and just turn it on a little, like taking 2 Advil. You clear 95% of the tumor. Once all the tumor burden is largely gone, you can put in a huge dose, and you don't get very much cytokine release, but you kill all of the tumor. So it's a way now to control this therapy. Again, we're going to go into patients, we're going to dose patients in 2019. And I think we're very soon going to enter into an agreement with a major pharmaceutical partner to actually take this approach and move it into solid tumors and use this now to treat a whole range of solid tumors like breast cancer, prostate cancer, pancreatic cancer, and others. I should also point out, because you can dose control this, it looks like you can improve the efficacy because you get a better what's termed memory response. And you only need 1 CAR T cell for all cancers. Now, we also have earlier stage programs. When viruses come into your body, they turn on signaling pathways to tell the immune system to kill virally infected cells. So now we're making small oral drugs that turn on those signaling pathways and tell them now to kill cancer cells. So we think, again, using the immune system will be an important way to treat cancers. We also have a large effort going on in regenerative medicine. For instance, osteoarthritis. 30 million Americans have osteoarthritis. When I get off my bike on Sunday morning, my knees hurt, okay? So we're clearly working on osteoarthritis, okay? It's a good thing about being a chemist. If you work on something at a young enough age, you might be able to do something about it, okay? The way you treat osteoarthritis is you take painkillers until you can't stand the pain anymore, and then you have your knee taken out, okay? And an artificial knee put in. If you want to treat osteoarthritis, what do you want to do? You want to put cartilage back, right? You've lost the cartilage, you just want to put it back. Where does cartilage come from? Well, it comes from a cell type called the chondrocyte. Where did the chondrocytes come from? They come from a stem cell in your knee. Everybody has them in their knee, called an MSC, a mesenchymal stem cell. So why not just take a mesenchymal stem cell, put it in one of those little wells, screen for molecules that turn it into a chondrocyte? If you turn it into a chondrocyte, it'll make new cartilage. So what do? You just look for cartilage in the little well, okay? We found that. We found the molecule. That's what you want to make. See if it works. So we put this into rodent and canine models. Here's a normal knee. Okay, if you destabilize this knee, you get what looks like an osteoarthritic lesion. If you treat this with a small molecule, you can't hardly tell the difference between the normal knee and the treated knee. So we now, actually, this is probably the first example of going from an idea in the lab all the way to a molecule. We're actually right now recruiting patients. I'm holding one slot for myself. And this could be the first disease-modifying therapy for osteoarthritis. A lot of people also have intestinal health issues, okay? And what happens is the intestinal barrier between all the microbes in your intestine and your body breaks down. And you get inflammation, cytokine leakage, and so forth and so on. And it's because your intestinal barrier is compromised. Well, there's a natural peptide that repairs that barrier, okay? It's called GLP-2. It's very similar to GLP-1 that some of you might take for type 2 diabetes. The problem with that molecule is it's very short-lived and very unstable. So we used simple chemistry to engineer a stable form that was long-acting and asked whether that simple peptide, its normal function is to repair the intestinal barrier, okay, and prevent cell death. Well, if we give a long-acting version, stable version, will that repair the barrier? And for instance, Crohn's and ulcerative colitis. So you can take a normal intestinal epithelium in a rodent model that looks like this. If you compromise the barrier and try and recapitulate an IBD-like phenotype, what you see is you can clearly just visually see the difference. This is a damaged barrier. If you treat with a simple engineered peptide, chemically engineered peptide, you can actually see you've almost completely restored the topography of a normal intestinal epithelium. So we think this could be a really interesting therapy to actually repair damage in Crohn's and ulcerative colitis. Another degenerative disease is MS, multiple sclerosis. Okay, this is an autoimmune disease where your immune system misfunctions and starts attacking your own body. And it specifically attacks the myelin sheath that coats the axons that connect the neurons in your brain. So the axons are like wire, the myelin sheath is like rubber insulation. You take the rubber insulation off, your brain shorts out, you have MS, okay? How do you treat this disease? The treatment today, the best treatment is to suppress the immune system, which isn't a good treatment. All you're doing is slowing down the disease progression, and it's potentially not safe. Simple solution to reversing this disease is to put more myelin back on, right? That's what's coming off, put it back on. Where does myelin come from? It comes from a cell type in the brain called an oligodendrocyte. Where does that come from? It comes from a stem cell we all have in our brain called an OPC. Just let's take some OPCs, put them in a well, screen for molecules that turn them into oligodendrocytes, and you'll make new myelin. It's that simple. So we did that, and it actually works. Okay, you find a molecule that makes new myelin, you put it into 3 different models of MS, and in each case it remyelinates and actually repairs the damage. What's interesting about this is if you slightly suppress the immune system and you repair, it will be the best-in-class therapy for treating MS patients because you're repairing damage and preventing damage. And what's really interesting are both of these treatments in 2019, drugs are going to be generic. So this could not only be the best-in-class therapy for treating MS, it could be the lowest cost therapy for treating MS as well. And that's a huge challenge. Another clear direction people are taking in trying to treat neurodegenerative disease now is realizing that there's a huge inflammatory component in neurodegenerative diseases like Alzheimer's and Parkinson's and ALS and others. So you really want to treat the inflammatory component of these diseases in the brain. How do you do that? Well, it turns out there's a naturally occurring protein called GM-CSF that upregulates a different kind of immune cell called a regulatory T cell that suppresses inflammation. So it does the opposite of what the cells are you're using to kill cancer cells. So if you can upregulate these regulatory T cells in the brain, you can actually fight inflammation. And people have shown when you give GM-CSF to a huge number of animal models of neurodegenerative diseases that it actually ameliorates disease. More recently, there was a paper published in Nature where people put GM-CSF into patients, Parkinson's patients, and actually saw they saw an improvement in motor function in a phase 1 clinical trial, which is really exciting, okay? The problem is this is a naturally occurring protein. It's got a very short half-life. So to get enough of it into the patient, because it goes away so fast, you have to give the patient a huge amount because it's gone so fast. And by giving the patient a huge amount, you cause a huge problem of side effects, okay? So what you really want to do is make a therapy that you take once a month and that has kind of a constant level of exposure, way below the safety level, but above the efficacy level. So again, we use technologies that were built at Scripps to engineer antibodies that have these activities. And antibodies have a huge advantage because they have a long half-life. And you can see here in this model, this PTP model of Parkinson's, that these engineered antibodies work far better than the GM-CSF that was put in the clinic. So we're partnering with the people who did this clinical study with this better drug that we think will have improved efficacy and safety. Now, we also have heard about the opioid crisis. And how do you deal with that? Well, you can make a different generation of opioids, which isn't so easy to do. Or maybe you can actually make an opioid vaccine. So you actually create antibodies now that don't recognize foreign viruses, but recognize heroin or fentanyl and actually pull it out of the body, okay? So these antibodies have actually been generated at Scripps, and in non-human primate studies, they actually reduce heroin potency and attenuate behavioral effects of the drugs. So this could be a very interesting way to begin to treat drug addiction. HIV. You know, patients who have HIV now can take heart therapy and they can live 30 or 40 years, whereas they used to live 5. But the problem is they always have HIV. It's not a cure. The HIV hides in cells in your body in reservoirs. So the minute you stop that therapy, you get a relapse of HIV. And it's not a particularly healthy therapy. So how do you eradicate, how do you cure somebody from HIV? So at Scripps, the structural biology group is looking at antibodies that are isolated from patients that are broadly neutralizing to the virus. These viruses change so fast that they change faster than the immune system can deal with it. So people have collected all of these different neutralizing antibodies and now use structure to come up with a consensus antibody that should be broadly reactive against all HIVs. And that, when you combine it with what's called a TLR receptor, okay, that activates innate immunity, looks like in monkey models that roughly 45% of the monkeys that receive both of those drugs, if you take them off of heart therapy, the virus doesn't rebound. So this could be actually a cure, or go in the direction of beginning to cure a large number of patients with HIV. This is being pursued in collaboration with Gilead. We're also looking at childhood diseases. Prader-Willi is a really terrible disease. It affects cognition, metabolism, and so forth. So the people in Scripps, Florida have actually found a molecule that works in the best animal models of Prader-Willi syndrome and are working towards taking that into the clinic. We're working very closely with the Juvenile Diabetes Research Foundation, not to treat diabetes. I mean, still today, kids are being treated with insulin, okay? They don't have beta cells. It's better to prevent the destruction of their beta cells, okay? So we're working with JDRF to find molecules that actually protect the beta cells in these kids before they're destroyed by the immune system, which would be disease modifying and potentially curative. We're also working on diseases of the developing world. We have a large collaboration with the Gates Foundation. And it focuses on diseases that kill more people than any other diseases we know of, okay? Malaria and TB TB kills 3 million people a year, okay? There are no cures, okay? And some of these diseases are persistent. So we've been working very closely with them. We actually already have drugs for persistent TB in the clinic in South Africa, as well as drugs for cryptosporidial infections. Interestingly, with the Gates, and we're working on worms, and we're working on HIV and other third world diseases. Interestingly, with the Gates Foundation, we made a collection of molecules of every drug that's ever been put into human beings. Okay, it cost about $20 million, but Bill was very generous and helped us out with that. Interestingly, many drugs have effects in more than one disease. So AZT, which was the first major therapy for the treatment of HIV, was developed as a cancer drug. Thalidomide, which was the first successful drug to treat multiple myeloma, okay, was developed as a behavioral drug, okay? So the idea now is if you have a disease, actually, why don't you just screen every known drug that's ever been put into people? And if one of them works, we already know it's safe. So you can actually go really quickly to the clinic. So the Gates Foundation asked us to work on cryptosporidium. It's the major non-viral cause of childhood diarrhea. About 100,000 kids a year die from cryptosporidium in the third world. So we started working on it 2 years ago. We actually, in one of those wells, put cryptosporidium infected cells and then screened the known drugs collection for a drug that would kill. Okay, and it turns out we found a drug that Novartis used to treat leprosy 30 or 40 years ago, okay? And it killed cryptosporidium far better than anything anybody knew. So within 2 years, we went from starting this project to a drug in people, okay? And so this is a really valuable way to accelerate drug discovery. In just a few more vignettes, it's really hard to make vaccines against some of these diseases like malaria and tuberculosis, and in fact, HIV. But we have really potent drugs that work against these diseases. What if you could alter these drugs such that instead of having them take them once a day, you could take them once every 4 months? That's almost like a vaccine, right? Every 4 months, I take a shot of a drug, it lasts for 4 months, prevents me from getting malaria, okay? So we actually have developed these now, and in collaboration with the Gates Foundation and Medicines for Malaria Venture, we're hoping to put these into human trials next year. We're doing the same thing for HIV. So you get a shot every 4 months, and you're protected from HIV. It's not a normal vaccine, it's a drug, okay? You can even take this a step further. What if you took a drug that every time a mosquito bit you, it died? Okay. That's an interesting idea, okay? So the Gates folks modeled this, and with potent enough drugs, okay, if you have a drug that lasts 3 or 4 months, it's predicted, based on modeling, that you'll decrease malaria by 90%, which is pretty impressive, okay? So we collaborated with DuPont. DuPont was terrific. They gave us their whole insecticide collection. And we found an insecticide that actually, it looks like it has a 3 or 4 month half-life. And it potently kills mosquitoes that carry malaria. It kills the mosquitoes that carry Zika, too. Interestingly, when we investigated this further, it turns out that drug, how many of you guys have dogs? A lot of you guys, right? You give them flea and tick? It's flea and tick, okay? That's the drug. It's the drug you give your dog every 3 or 4 months. Okay, well, it turns out if you take it and a mosquito bites you, it'll probably die, okay? So Mr. Behringer was kind enough to convince Boehringer Ingelheim to actually let us use an analog of that drug. And so we're actually, again, working with MMV to test that as an anti-malarial. But it turns out, if you ask a simple question, How about Lyme disease? Lyme disease we've all hearing about on the news. It's growing in the Northeast like crazy, the Midwest, the Bay Area. When you give your dog flea and tick, does it get Lyme disease? It's running around in the bushes and the fields and everything. No, it doesn't. So it turns out we know that that also kills the tick that carries Lyme disease before it passes Lyme disease to the dog. Yet we're getting Lyme disease and our dogs aren't, okay? So why aren't we taking this ourselves, okay? So again, we're actually working with folks to actually, if we can show this works in malaria, I think you could move it over to Lyme disease. You take a drug at the beginning of the summer and you're protected from Lyme disease. It's a good solution. And finally, STSI, the Scripps Translational Sciences Institute, has become part of Scripps, as has Caliber. Now they're all together, okay? All one institute with one board. STSI is focused on precision medicine, using all of the genomics and proteomics and metabolomics tools to understand how disease varies from individual to individual. This is called precision medicine. The answer is my disease isn't the same as your disease. Okay? And how do you begin to understand how diseases, to prevent diseases and treat diseases on the individual patient level? So what you need to do is gather a huge amount of information from the public and correlate that with disease progression and disease treatment. So the NIH gave Scripps a $250 million grant, which is the largest grant given by the NIH, to begin to collect this data, to really begin to understand disease at the level of individual patients, and that will certainly change the treatment paradigms we use. So how does all of this, so the model now is not only to do outstanding basic research, but to combine that with Caliber, which does translational research, and STSI that does precision medicine and genomic medicine. Have all of that within one institution, Scripps Research, that works together to really accelerate the impact science will have on the public health. And that's really the goal, okay? It's not to fund research for 30 years and ask, my kid still is taking insulin. It's like, can we actually make a cure for type 1 diabetes? And the other thing that's going to come out of that, and it's important, is that as you create these new medicines, you're creating those for the public wealth, but you're creating, medicines have huge value. And as we create these medicines and move them further along in the Institute is going to share more in the value creation process. And that's going to create huge resources that we can reinvest in basic research. So we're no longer dependent on simply government funding to move science forward, which is becoming more and more of a challenge as we move into the future. And so the goal, and I'm sure Bradley's going to quote me on this, is in 3 to 5 years, okay, that we'll have resources from all of these combined activities that will allow Scripps to be unique in the scientific world, to have enough resources so that if anybody has a good idea, no matter how risky it is, we'll have the funding to give to them. And if anybody has an idea for making a new medicine, we'll have the resources to actually begin to create and test those medicines, not only preclinically, but also in the clinic. So that's the goal, and that's what we're trying to do. And I think with some of the advances I've described today, hopefully, I think we're beginning to see light at the end of the tunnel. And I think if we can make this new model work, not only will it benefit us, but it'll benefit a huge number of other nonprofit institutions around the world. Finally, again, I'd like to thank you for coming here today and hearing about the direction we're taking Scripps. We're excited about it. I really want to acknowledge all of my colleagues at Scripps. Just a terrific faculty, hugely supportive staff, graduate students, postdocs are just amazing. I'd also like to point out the caliber of people who have done a lot of took the risk of joining a nonprofit drug discovery effort that had never been done. Many of them took pay cuts, but I think they're starting to see, the fruits of their passion and hard work. And I think together we're going to do something very exciting that could have a big impact. And I'd like to, if you have any questions, we probably have a few minutes for questions, and then I'd love for you to join my colleagues and I for a reception out front. Thanks a lot. Any questions? It's probably kind of an intimidating,
Fantastic presentation, by the way. Thank you so much. Have you thought about anything in regards to potentially rejuvenating cells or cutting back the time for most cells?
Yeah, so cellular senescence and rejuvenating cells, it's an interesting question. Can you people are trying to selectively kill cancer cells versus normal, and there's a reason for that, because you die of cancer. Trying to kill senescent cells versus normal is because you want to live longer. It's a higher bar. It's probably more complicated, but it's a really exciting area to move into. The problem is if a mouse lives longer, does it really mean you're going to live longer? So it's hard to test those ideas, okay? We're starting out with the easy stuff, okay? What else? Any other questions? Yeah.
I recently learned that you can cure a cat of diabetes. And if that is possible, I know cat biology is quite different than human. Is there a way we could use the technology that they use to cure their disease to help in human diabetes control?
Yeah, that's an interesting question because, one of the big challenges in developing new medicines is how do you know it works until you put it into people, okay, and test it? And so people use animal models all the time, okay? And the question is how translatable is if you protect beta cells in a cat, are you going to protect them in humans? We have cured more mice of cancer than you can shake a stick at, okay? And how do you know if a mouse is depressed, okay? There's huge challenges there, and some of the things that, for instance, Kristen Baldwin is doing, where you actually take skin cells, you don't have to use a cat. You take skin cells from a patient with Alzheimer's, and you make the Alzheimer's neurons in the laboratory, and then you test directly on human neurons that have that disease, okay? So those are the directions we're going in, and that will really accelerate the ability to take a drug that works in the laboratory and move it into people with the confidence that you'll see something. Yeah.
The idea on that vaccine for the opioids,
right.
Would that affect then patients later if they had a pain issue that they couldn't take, or it would have no effect?
Well, it would work for, it would be, what you would do is probably make a vaccine against fentanyl and heroin or some combination thereof. If you give a different opioid, you would overcome that, so you could simply go to a safer opioid for the pain. But the idea is it would be a way to take people who are really addicted and take them off the drugs in a more benign way. That would be the idea. It's a pretty exciting approach. It's very different and innovative. And I think that's part of an advantage of doing, not only basic research, but also translational research, in an academic setting, because in general, academics take more risk than pharma. In general, an assistant professor, a young person, to make their career in science has to take risk and do something different. And big companies, a lot of times what you don't want to do is make a mistake. So it's almost the opposite culture. And today you want people to take risks to cure diseases, okay? But you also want those risk-taking activities to be cheaper and faster so you can fail quicker. And every now and then succeed, okay?
Does the FDA have a role in this? And do you see the future of medicine sort of eliminating the FDA's role?
Yeah, we clearly have to make sure that our medicines are safe, and there's a risk-reward paradigm that, people probably have a different risk-reward paradigm when they go into their car than they do when they take a drug. And so, I was talking to the head of R&D of a major pharmaceutical company who had a drug that would prevent viral diarrhea in the developing world. And a huge number of kids, I think maybe 500,000 kids die every year of diarrheal disease in the developing world. So if you gave this vaccine, you would probably prevent 490,000 deaths. But you can't develop it in the third world because there's a certain risk associated with it in the U.S. that's a very low risk, bar. Whereas, we wouldn't take it. If 1 in a million kids died from that vaccine, you probably wouldn't take it. But on the other hand, if you save 490,000 lives a year in the developing world, you would do it, okay? So there's this whole problem with risk-reward and how does that impact people's health. It's really not only a scientific issue, it's a social issue. And so I think it's an interesting problem.
Can you talk about where you think the sweet spot is for Caliber in the drug development process? It seems like you guys are running a lot of phase 1 and phase 2 programs. And then is the model then to then sell or license the drug to Big Pharma for them to do the phase 3 study? Or do you foresee Caliber going into phase 3 development as well in the future?
Yeah, some of the challenges of developing drugs is as you go into the later stage clinical trials, the costs become huge, okay? And there's no way, I think, a basic research institution, would be able to support those costs. So you need to partner with pharma, and pharma has a huge amount of expertise in running complex phase 3 trials. So really what I think we'd like to do in general is take these through safety studies to human proof of concept studies, show the drug works, and then partner with pharma. And really they have the resources and expertise to move it all the way through to approval so patients can use it. So it's a collaborative model. But the other part of the model is to collaborate with other institutions to start drug discovery projects. So ultimately what we'd like to do at Scripps is not only use the resources we have to accelerate discoveries that are made here at Scripps, but also collaborate with folks in the Bay Area, Boston, and other places.
So about the Bill Gates database of every compound that was put into a human body, that idea sounds pretty straightforward, but you said that $20 million went into it. How long did it take to make that, and what were some challenges involved?
It took 2 years to do it, because first of all, you had to figure out all the drugs that had been put into humans, and pharma doesn't necessarily advertise what molecules they put in for, intellectual property reasons. So it took a lot of bioinformatics and cheminformatics effort to figure that out. And then we had to figure out the syntheses of these molecules, okay? And then we had to make these molecules, okay? But the whole thing took about 2 years, start to finish. Costs about $20 million. And now what we're doing is we have Wellcome Trust visiting us in a few weeks. And what we'd like to do is work with them to make this collection available to the entire academic community. So anytime anybody has a question, okay, or sees a disease, or even goes into the hospital and takes skin cells and makes disease in a test tube for somebody with cancer, that they can test this collection against, those diseases. So we're working pretty hard to expand the access of this collection to the whole world. Well, I think, the faculty's around, the Caliber people are around, STSI, we're all one now, okay? And people would just, if you see somebody who looks like a faculty member, a little haggard and tired, okay? Or a graduate student or a postdoc who's more haggard and tired, okay? Feel free to ask them questions, and thanks a lot for coming.